Thank you.
Wait, did it start?
Yeah, it started. Welcome, kids. Today's pretty special because today we are going to finish our discussion of chapter 12, which is the last chapter we're covering this semester, which means that there are no more notes to take after this class. Next class on Friday, the day we head out off to Thanksgiving break, we are going to do a lab. There will be an online and an in-class part. We'll use the microscopes, and it will be related to what we're going to talk about first today. When we get back from Thanksgiving, we will make sure our video projects are wrapped up and done, and we will do some review type activities for chapter 12. And then on December 8th, which is that second week after we get back, your projects will be due on the same day we take a half test on only chapter 12. There will be a few classes, maybe 3 classes after that, and you will have some reading to do on viruses, but you will never be tested on it. I will simply just have you take notes on the reading, and I will grade your notes. That will probably be your first grade for next semester, but that will be your homework. And we will go over some kind of exam review things and focus a little bit more on the AP exam and some practice questions that you can work on for just kind of a final little bump to your grade. So, chapter 12. We've been talking about mitosis to this point, and we're going to continue talking about mitosis today. And just remember, we're essentially discussing how a eukaryotic cell, a cell with a nucleus, is going to separate out its chromosomes so that the new cells that are produced after cell division are going to have a full copy of DNA, which are the instructions that your cells use to do everything that they do. So we've talked about these different phases in the cell cycle and the circle of life of a cell. And so I wanted to show you some actual pictures, because one thing you'll have to do on Friday and something that you'll have to do on the test is look at either diagrams or actual micrographs, pictures under a microscope, of cells, and tell me what phase they are in. And that's a lot of what we're going to practice once we get back from Thanksgiving. And it's also what the lab on Friday is about. So what kind of cell do we have here?
Plant cell.
This is a plant cell. You can see the boxy outline representing the cell wall. This is the nucleus of the cell, as is indicated on the diagram. And I do have it labeled here at the bottom with the name of this phase. This is prophase, which is the very first part of mitosis, after the cell leaves a time in its life called interphase. So remember, most of a cell's life is spent in interphase, where it is growing and at some point preparing to divide by making a copy of each chromosome. We've already done all that in this cell, and this is the first part of mitosis, where the cell is going to start separating its 2 copies of chromosomes and dividing at the end into 2 separate cells. Now, a major event in this process is listed right here. The chromosomes, which are normally long and thin, are going to start wrapping around themselves like a thread being wrapped around a spool. And so they become shorter and thicker. That's what we mean by this chromosomes condensing. The chromosomes, which is the DNA, it's the packages that all your DNA is stored in, they become shorter, thicker, and actually visible under a microscope, which is what we're going to be looking at on Friday. So that's what a lot of this dark structure is here in the nucleus. This is you're seeing here the beginning of that condensation of the chromosomes. They're becoming shorter and thicker. All right? So that's prophase. Now we'll run back through all of these after we get through, just so you can kind of quickly see the difference in these phases. From prophase, we've got this stage, or the second part of mitosis, that we call prometaphase. Now, this you can think of as being the same cell. It's probably not the exact same one, but it's where the previous cell is heading. And if you notice, there is much more condensation that has occurred in the chromosomes. Okay? So between prophase and prometaphase, your chromosomes will completely condense and become basically as short and as thick as they ever will, meaning they are at their most visible and most condensed point. And you can kind of see that if you kind of look as I toggle between these two pictures. There's less condensing of chromosomes here, and then more condensing of chromosomes in this second part known as prometaphase. Now, for the most part, these chromosomes are still kind of jumbled around and randomly situated throughout the nucleus. It's also at this point that the outer layer of the nucleus, and you cannot really see that in this image here, but this is something that is known to happen. The outer layer of the nucleus, that we call the nuclear membrane or the nuclear envelope, it is dissolving. It is breaking apart and going away. So what's the next phase? Metaphase. Good. Metaphase, a lot of times what you want to do is associate meta with middle. That's a good word association for mitosis. Here, and it doesn't really look as clear in actual pictures as a lot of times it's drawn in diagrams, but here you essentially have the chromosomes lining up along a central axis. Now, I wouldn't say that they're all perfectly in line or aligned with one another, but you can see that there is an arrangement of chromosomes around a central axis, which your book refers to as a metaphase plate. But the chromosomes are lined up in the middle. Now, there are other events that occur during metaphase that you could not glean from just this image. Things like the fibers that are being produced are actually attaching to the chromosomes at this point. But we'll get into a lot of the specifics of those other events later. For the most part, you need to be able to see this cell, see the chromosome arrangement along the center, and identify it as metaphase. What's the next phase? Anaphase is your next phase. And just like a lot of times you think of meta and middle, I like to think or associate ana and apart. It's a nice little alliteration to help you. Because anaphase is where you begin to see, and I know this is not a human cell, but if this were a human cell, you would see 46 chromosomes moving in one direction and 46 moving in another direction away from each other. The sister chromatids, the duplicated copies of each chromosome, are going to be moving apart in anaphase. That's where you see separation. One set heading to one end of the cell, the other set, complete set, moving to the opposite end. Yes?
Now, like, anytime, can we see, like, the cell plate, like what we talked about yesterday, like, when, the vesicle comes in?
I will show you in the next slide the cell plate that is used to kind of construct a new cell wall in plant cells. That's a great question, and you'll be able to see it right here in telophase. It's a well-timed question. Telophase is the end of mitosis. Okay? So what we see here is that the complete set of chromosomes that was moving to one end of the cell is now going to have a nuclear envelope form back around it. You really can't see that, but you can see that this is a new nucleus. And the other set of chromosomes has now is going to be enveloped by a new nuclear membrane. You essentially have two separate cells at this point because the cell plate that is constructed by the plant cell between these two nuclei, and it is the division between these two new cells. Yes, ma'am?
Do they have the same cell wall?
This will eventually be a cell wall made of the same material that functions the same
Like,
way
do they
as any of
share
the other.
it? Do they share it?
Oh, I see what you're saying. That's a fantastic question. I don't know what characteristic delineates or forms the boundary between like this plant cell and this plant cell. I don't know if there's a dividing line because it's kind of like the wall of the classroom. That wall is essentially, it's the outer wall of the building and the wall of the room. So I don't know, or say a wall between two rooms. I don't know that you would really have a marker that indicated where the wall of Miss Haynes's room and my room begins and ends. I guess it's one wall.
Okay.
There could be something, but I don't really know. I would assume the material of the cell plate serves as kind of the dividing line, but that would be more of a guess. It's a great question. All these great questions and answers on this chilly Wednesday morning. Now, that is mitosis. Now, the building of that cell plate and the cell wall that will form around it is not really considered part of telophase. It happens during telophase, but we really refer to any dividing event as cytokinesis. So when you hear the book reference cytokinesis, that's kind of what events take place to physically separate two cells from one another. And it happens around telophase, but is considered to be kind of its own unique event. When we say words like prophase, prometaphase, metaphase, anaphase, telophase, we are really talking about what's happening with the chromosomes and how they are being segregated into different ends. And then cytokinesis is how you separate those two cells. So again, in general, looking at these pictures, you have the start of the condensing of chromosomes. You have the chromosomes fully condensed. You have the chromosomes lined up in the middle, pulled apart, and then essentially new nuclei forming, each with the correct number of chromosomes. All the chromosomes in this cell are duplicates of the chromosomes in that cell. And we have now gone from one cell to two, and that's cell division. In eukaryotes, which are more complex and makes mitosis more complicated than this next quick topic that I want to talk to you about, which I have titled here, binary fission.
Wait, so cell division only happens in eukaryotes.
Cytokinesis, well, cell division happens in all cells,
Yeah.
whether they're prokaryotic or eukaryotic. Eukaryotes utilize mitosis.
Okay.
Now, in prokaryotes, what do you not have?
Nuclear.
You don't have a nucleus or a nuclear membrane or envelope. You also, in prokaryotes, typically don't have what you would call chromosomes. Usually, all of the DNA is going to be on one circular loop of DNA, a single package, if you will. We don't even really use the term chromosome. You will see the term plasmid used in your book. You may have already seen that term, but if you haven't, you will see it. So here we have a little bacterium with no nucleus, and you have just the DNA in basically one package, not 46 different packages called chromosomes like in human cells, but just one either circular loop of DNA or one package of DNA. And I guess we could have talked about this first, but your book didn't go in that order, and that's fine. But I wanted to stick with the book order since we're reading through these chapters. But essentially, the one package of DNA that a prokaryote will have is going to duplicate itself. Now, it starts this replication process at what we're calling the origin here. And as soon as you start duplication, one copy kind of arranges itself to one side of the cell, and the other copy is going to arrange itself towards the other side. And then once the copying of DNA is complete, then you typically have some sort of cytokinetic, some sort of cytokinesis occur in which the two cells pinch off from one another. I'm not sure, it probably depends on the bacterium whether you have more of a cell plate or a cleavage furrow that forms. Some bacteria have cell walls, some don't. Some have weird slime layers that are kind of like a cell wall, but not a true cell wall. So there's a lot of diversity in the world of prokaryotes. But essentially, in prokaryotes, which are much simpler, much smaller, and always single-celled, you're going to just have the one copy of DNA, or the one package of DNA, copy itself. Each copy is going to kind of lean towards one side, and then a division takes place, and it's a much simpler process. You don't have to make sure you get this copy of this chromosome to one side, that copy of that chromosome to one side, dozens of times, however many chromosomes the organism has. And this is referred to as binary fission. No mitosis is necessary. You don't have to segregate a bunch of different chromosomes. There's not a nucleus that has to dissolve and then reform. You just simply have a copying of DNA, one copy to each side, and a division of those cells. Much faster. By the way, what's the purpose of cell division in a single-celled organism? What's the result? Why is a single-celled organism going to divide? To what ends? It's for reproduction. This is just how you get more individuals in a single-cell population. Whereas in humans, we engage in cell division for purposes of growth, increasing complexity, and to repair lost or damaged cells. All right, so there's 2 more topics covered in this chapter. I'm going to spend just a little bit of time on this topic. Your book goes into a bit more depth, so you want to make sure to read about this or focus on this when you read about it. And then we'll wrap up the chapter with the last topic. But in general, and I'm not too concerned about the details because there's a number of chemicals that go into this. In general, the process of going through interphase, which remember is subdivided into these other phases, G1S, G2 phases. Interphase, mitosis, interphase, mitosis, interphase, mitosis. That cycling that a cell goes through during its life is controlled, or should be controlled, by chemical signals. And this is kind of where we go back to chapter 11. It's why chapter 11, which talks about cell communication and signals and responses, is before this chapter. Because all this mitosis stuff, whether you're in interphase and when a cell gets the signal to enter mitosis, it is all driven by different chemical signals. There are chemicals, and again, your book talks about these. We have what are called cyclin-dependent kinases, and this other chemical known as cyclin. These chemicals build up. They will actually combine once their concentrations are high enough. That is the signal the cell needs to go through mitosis. And once mitosis is over, some of these chemicals break down, and you get a signal to the cell to enter back into interphase. Enter into interphase. So whether the cell is in interphase or is deciding to enter mitosis and divide by the end of mitosis is dependent on a few different signaling chemicals. Some chemical signals. You can see here concentration levels building up, peaking during mitosis, and then those concentrations crash and then flatline for a while until they build up again and send the signal for the cell to divide again. I mean, that's just how the cell, I don't like using the word knows, but it is how the cell is directed through these different phases of its life. So the main point here is that, a cell doesn't have a brain per se. So it doesn't know or decide when to divide and when not to divide. It's driven by a few different chemical signals that accumulate and at a certain critical concentration sends the cell into a phase. And then a lot of times it's the degradation of those chemicals that cause that cell to end the phase and enter into another part of its life. So it's chemically driven. Your reading goes into the chemical details. And we can review some of the primary chemicals later for this process. But I want to finish, and I want to give you time to work on your projects. So the chapter ends with a discussion of cancer, and specifically breast cancer, and how it's connected, how cancer in general is connected to this concept of cell division, mitosis, and chemical signals that cause cells to enter into mitosis and to divide. You've all heard of cancer. It is typically going to be initiated by some kind of mistake in your genetic code. Now, your genetic code can be altered in a cell based on exposure to radiation, based on your exposure to certain chemicals, or maybe through just some kind of faulty process that accidentally occurred. And so here we have, tissue within the breast, and they point out the tumor that is forming here. Now, a tumor is a cluster of cells that are all considered to be cancer cells. And this can occur in any part of the body. Now, as it says here, a tumor comes from a single cell. So how many cells did I say were in your body?
Trillions.
Yeah, the number is probably around 1 trillion or more. Or maybe a little less, but yeah, the number is something like trillions of cells. One cell loses its ability to regulate its division cycle, its cell cycle, and that one cell can turn into two cells, both of which have the same problem. They don't know when to stop growing and dividing. Those two cells turn into four. Those four cells, all four will split in an uncontrolled way because they are not regulating themselves properly because of some issue in the DNA. And those four cells become 8, 16, 32, 64, 128, 256, 512, 1024, 2048. And it keeps going until you have thousands of cells in a cluster, all duplicating their DNA, which has a mistake in it that is causing them to grow at an out of control rate and to divide more often than they should. So that's what a tumor is. It's just a mass of cancer cells. They are your own cells, just not functioning properly and growing too fast and dividing too frequently. So as those cells continue to do that, the tumor can get larger. A big problem with tumors is that they steal nutrients from cells around them that are actually trying to do their job and keep you alive. They also can put pressure on tissues and organs around the tumor, like physical pressure, which can cause pain, but can also cause organs to not function correctly. Left unchecked, these tumors can actually kind of interact with blood or lymph vessels. And what can happen as a result is some of these cancerous cells can actually dislodge from the tumor, enter into either the bloodstream or what we call your lymphatic system, which is a system we really don't talk about, and it's not discussed very often in many courses in biology, but it regulates kind of fluid buildup and fluid collection in the body. But if you have vessels that are transporting anything from one part of the body to the other, and a cancer cell ends up in them, that cancer cell can travel to another part of the body and start the tumor process all over again. That cancer cell can travel somewhere else. If it gets stuck in one place, it will start dividing there, and a new tumor will grow. And we have the layman's term for that is that the cancer has spread. The technical term is metastasis. We say that a cell or that the cancer has metastasized. That is the technical term for cancerous cell breaks free from a tumor, travels to another part of the body, and starts another tumor. And that is the real danger of cancer, is a situation where you have multiple tumors maybe tumors growing in places that aren't so easy to remove. And that's, depending on how long that process goes on, you can find yourself in getting into trouble with with the cancer that you might have. So a lot of times you'll hear this term early detection of a tumor. Especially, they have breast exams, mammographies where you can detect these tumors. They have, many different diagnostic processes. Like with prostate cancer, they can do a blood test and see if certain enzymes in your blood are at a higher or lower level than they're supposed to be, and that is an indication you might have a certain type of cancer. But if you can find a cancerous growth early enough, it's very likely that you can remove it and treat it before it metastasizes. And if you can, then you can, remove that cancerous tissue from the body and you're fine. And I don't want to take a dark turn or to be pessimistic about this, but remember, how many cells do you need to have to start a cancerous tumor?
2.
One cell out of trillions. And you're constantly making new cells. You're constantly being exposed to things in the environment. Cancer is kind of a situation that if you live long enough, you just get some form of cancer at some point. Again, I'm not trying to sound pessimistic, but it is kind of a reality of life. And what I'm really saying is that it's important, once you get older, that you go to the doctor every year and get checkups and have your doctor screen you for things like this and catch it early enough. And your book talks about cancer at the end of chapter 12, and make sure you read it. So, at this point, and really everything we're doing between now and Thanksgiving break will be working on your video project and the lab activity on Friday. So we're going to stop the video, maybe the last video, one of the last videos of the semester. We're going to stop the video. We're going to stop the video. Bye.